Assalamualaikum warahmatullahi wabarakatuh. Okay, today I want to explain about running a protein gel. Okay, first part is collect the cells. Okay, first, set the temperature of the heat block to 95 degrees Celsius. Okay, next, set the P200 micropipette to 30 microliter. Then, attach a P200 tip and move the micropipette so its tip is in the wild-type cell culture. Draw up the wild-type cell culture by pressing the plunger to the first stop, immersing the tip in the solution, and releasing the plunger slowly. And then close the wild-type cell culture. Move the micropipette so its tip is in the wild-type cell tube. Use the eject button to eject the tip into the trash. And then repeat the procedure to dispense 30 microliter of the pancreas cell culture into the pancreas cell tube. Next, repeat the procedure to dispense 30 microliter of the EDIT4 cell culture into the EDIT4 tube. Next, repeat the procedure to dispense 30 microliter of the EDIT7 cell culture into the EDIT7 tube. Okay, the second part is add sample buffer. So first, set the P20 micropipette to 10 microliter, and then attach a P20 tip to a P20 micropipette, and move the micropipette so its tip is in the 4 times SDS sample buffer. Draw up 10 microliter of 4 times SDS sample buffer by pressing the plunger to the first stop, immersing the tip in the solution and releasing the plunger slowly. Next, close the 4 times SDS sample buffer. Pop-up question. What is the purpose of adding SDS to the cell cultures? A. SDS solubilize membrane proteins. B. SDS breaks down cell membranes and releases the cell content. C. SDS destroys protein. D. SDS unfolds proteins and covers them with negative charge. Negatively charged proteins can be separated in a protein gel according to mass after applying an electrical current. E. All except C. F. All of the above. So the answer is all except C. Next, move the micropipette so its tip is in the wild type cells tube. Dispense 4 times STS sample buffer into the wild type cells tube by holding down the planner until it reaches the second stop. And then use the eject button to eject the tip into the trash. Repeat the procedure to dispense 10 microliter of 4 times STS sample buffer into the pancreas cells tube. Next, repeat the procedure to dispense 10 microliter of 4 times STS sample buffer into the EDIT 4 tube. Next, repeat the procedure to dispense 10 microliter of 4 times STS sample buffer into the EDIT 7 tube. The protein ladder already contains STS sample buffer. The third part is denature the protein. So, move all of your 4 sample tubes to the heat box. And then set the heat block timer to 10 minutes and press start. And then remove all of your sample tubes from the heat block. The next part is centrifuge the samples. First, open the microcentrifuge lid and move your 4 sample tubes to the microcentrifuge. The protein ladder does not need to be spun down. Place the 4 tubes of fill receptacles as symmetrical and close the microcentrifuge lid. Press the pulse button to briefly spin the liquid to the bottom of the solution tube. And then remove the tubes from the microcentrifuge. The next part is load the gel. Place the polyacrylamide gel in the electrophoresis box. And then fill the gel cassette with one-time running buffer until it covers the wells of the gel. Next, remove the comb from gel cassette. And then fill the outer gel electrophoresis tank with one-time running buffer. Set the P200 micropipette to 20 microliter and attach a P200 tip. Move the micropipette so its tip is in the protein ladder tube. And then, draw up the protein ladder by pressing the plunger to the first stop, immersing the tip in the solution and releasing the plunger slowly. Next, move the micropipette so its tip is above the first lane of the protein gel. Dispense the protein ladder into the first well by holding down the plunger until it reaches the first stop. Use the eject button to eject the tip into the trash. Pop-up question. How should your micropipette samples into the wells? A. Quickly so the proteins go down in the well. Slowly to prevent it spilling over into the next well. C. Slowly but move the plunger up and down to mix. The correct answer is B. Slowly to prevent it spilling over into the next well. Next, repeat the procedure to dispense 20 microliter of the positive control white blood cells into the second well of the gel. Next, repeat the procedure to dispense 20 microliter of the negative control pancreas cells into the third well of the gel. Next, repeat the procedure to dispense 20 microliter of the EDIT cells into the fourth well of the gel. Next, repeat the procedure to dispense 20 microliter of the EDIT7 cells into the fifth well of the gel. The last part is conduct protein electrophoresis. First, place the cover over the electrophoresis box, and then select the red and black cables and connect them to the red and black connection points on the power supply, and the red and black connection points on the lid of the gel electrophoresis box. Turn on the power supply, and then set the voltage to 150 volts. Set the timer to 60 minutes. and then start the power supply. Wait until the time has elapsed. Pop-up question. How do you know if the gel has run for long enough? A. The diaphragm is missing. B. The diaphragm is at the top across the wells. C. The diaphragm is at the bottom of the gel. The right answer is C. The diaphragm is at the bottom of the gel. Next, turn off the power switch and then unplug the electrodes from the power supply. Carefully remove the cover from the electrophoresis box to inspect your gel.